For cfNA fusion fragments. The talk is presented by Xiao Zhang. And I remind the speakers that they have 20 minutes, so I'm going to be sitting in front of the screen, and when there are only 3 minutes left, you should see me saying something like 3. At 20 minutes, I will come out here with you. Thank you.
So I think I'll do a quick presentation. So I think Christina emphasized the importance of early detection. So I'm from Grail. We are a company doing the exact early cancer detection. This is just one aspect of work we are doing from actual R&D side. So what is the problem here? This is a nice figure from the TCGA. What this shows is from the healthy situation, you can see the gene A and gene B. They are being transcribed independently into 2 different genes. And in the case of the segmental duplication, inversion, or deletion, you can see there's a break in the exon or at the exon boundary or intron region of gene A and gene B. Then the 2 transcripts get fused together. As an outcome, there's a new RNA being formulated, and many of these translated to a protein. So in 2008, we have known that TMPRSS2 fusion are detected in 50% of prostate cancer. And 10 years later, we have a lot of TCGA data. And as the fusion is identified to be prevalent in this data set, this group identified about over 25,000 fusion events in over 9,000 tumor across 33 cancer types. So importantly, fusion could be a driver events. It drives about 16% of the cancer cases in 1% of them. It's a solo driver events. So actually, most importantly, some of these fusion events are druggable. That's your ultimate goal. You want to treat this cancer when they have been detected. So 6% of them involving these gene events already have the drug. So as you can see, if you Google this fusion tool online, you can see like there might be 30, 40 tools already available. Why do we write a new program? This is a nice figure from the Star Fusion paper, which demonstrated the runtime and the memory usage for the existing fusion detection methods on this very tiny small data set. This is a 60 paired and cell line data sets of 20 million rays only. You can see that we only consider, let's say, if Star Fusion is running a reasonable time frame, and there are assembly-based methods such as Jaffa hybrid. This on a larger scale data set will never work. Secondly, there are unique properties of the cfRNA and cfDNA data. Due to the fragmentation pattern, this data set centered around 167 or 166 base pairs. It creating artifacts in the data like overhangs. What does that mean is basically if your read length, let's say typically 150 bases, if your fragment size is less than that, then the sequencing data reads into the primary region and create overhangs. I'll get into the details later. So in the circulating DNA RNA, so the CT stands for tumor fragments. The proportion is typically very low, as low as 0.1%. So because of this, we need to sequencing this like 20 times larger in order to find the tumor fraction. Then there's a specialized, because of this data, the property of this data, there are a lot of highly PCR duplications, so we tag this individual reads with unique molecular identifiers. So they have this special tags. So we have the rationale before we developing this tool. We consider alignment-free k-mer-based strategy would not be plagued with soft clipping events, which is confounding the existing aligners. So the majority of data, because the majority of data are actually not Fusion fragments, we think it's possible to rapidly filtering out the majority of the data. Once you do that, you can use more sophisticated algorithms to further filtering down to the biological relevant events. So there are two types of data. One is the cfRNA we consider might be more appropriate to identify the fusion events, but majority of the panel right now is based on cfDNA data, and over half size of the panel are dedicated to the fusion. So next question is how do we implement? So this is the overall workflow. Consists of three parts. I will go into slightly detail later, but our tool consists of major two modes. One is the target mode, one is the de novo discover mode. So we consider majority of the time the investigators or the companies are only interested in the target mode because we've already known what's the driver fusion events, what's already fusion events that has been identified to be relevant. So intuition behind this problem is as shown on the left figure. On the top, it shows the transcripts consists of G1 and G2. XB is the breakpoint. On this transcript, you are going to sequence it. Then it mainly consists of three different fragments. One is the fragment does not overlap with this fusion joint point. So R1 and R2 spanning do not overlapping with this breakpoint, but the other case is one of them overlapping with this breakpoint. Then the third one is both overlapping with this breakpoint. This translates mathematically to two problems. One is you expect alpha fraction of R1 and R2 combined map to G1 and G2. To account for sequence errors, we typically choose this alpha to be less than 100%. So the secondly, R1 and RB would share a minimum of beta basis on both sides of the fusion junction point. So traditionally, other methods just choose 25 base pairs to improve the specificity. So as I mentioned, the method consists of three parts. This is the input part. For the reference curation for the cfRNA data, we just took the existing transcriptome file, plus we panned in the exome boundaries with a fixed number basis. The reason to do this is this can reduce the false positives of reads spanning intron-exon junctions of the unspliced nascent mRNA. For the cfDNA data, we simply take the entire genic space, which is about 1/3 of the genome. So another step we applied here is we pre-process the input into a k-mer hash table. It's basically simply a k-mer to which gene occurs. You can note that completely we forgot the positional information. So in the target mode, we curated about 655 the current fusion gene known pairs from the COSMIC database and some curated additional known targets from the literature. So the list input consists of a list of compressed FASTQ files. If the sample is sequenced, let's say in 8 lanes, you get 8 pairs of FASTQ files. We have to emphasize that here, because of the gzip format, the algorithm is actually limited by the I/O itself. So typically, if you have a single paired FASTQ gz file, you can do a Linux command to split into multiple ones to increase the performance. So for stage 1, simplistically speaking, we process each read pair independently. So this can be highly parallel. What we do step by step is we first convert the read pair to a fragment, then drop low complexity fragment, then identify all candidate gene pairs that support the fragment via the k-mer hash table. Then we infer the genes, each gene pair that best supports the fragment, which I will explain later. So there are some practical considerations. One consideration is what size of the k-mer you are choosing. The other consideration is if the k-mer is highly frequent, then what do you do with it? So empirically, we have done some study which showing that the k-mers ranging from anything between 17 mer to 19 mer is sufficient. And highly frequent k-mers, k-mer greater equals that 5 occurrences in the gene, we can safely drop it. So there are some details about the stage 1. There are 2 points I want to emphasize here. The remaining you can get from our paper. So this is a fragment generation. As I mentioned, the majority of the fragment falls into case A and case B. As you can see, because our fragment is centered around 166 bases, so many of them are lower than 150, which is the typical read length. So this reads into the overhang region, which is typically a primary region. So you need to just chop it to form the final fragment, which is the intersection between R1 and R2. So in rare cases in our data, R1 and R2 do not overlap. In order to convert, we artificially convert this still to a fragment by reverse complement R2 to concatenate with R1. The other factor is I talked earlier, we kind of infer the gene pairs that best support the fragment. What does that mean is there are 2 steps. One is we approximate the coverage of the fragment F by any gene without the positional information. What does that mean is, give you an example, let's say G2, there are positions that you know a particular k-mer is from G2, but you don't know where the position is, and you just simply identify all the positions that belongs to G2. You just blindly concatenate. So that, let's say G2 covered this particular S4, S5, and S6 region. Then G1 covered another subset of regions. These empty regions are likely due to sequencing error and other artifacts in the data. I only illustrated G1, G2. Here, typically, you see a long list of genes. Then this problem turns into a max cover problem. You basically identify 2 genes that best cover these fragments. Then on the stage 2, we just choose the empirical set of criteria to improve the specificity, specifically low complexity sequence, nearby genes, PCR duplicates, and genes with 2 million partners. So I have to emphasize that because of the nature of the development of our tool, for the first step, you actually have the very high sensitivity and specificity. The size of the data coming out of stage 1 is already very small. So if you are not satisfied with the empirical criteria we listed here, you can combine the data from stage 1 to other more sophisticated filtering methods like implementing star fusion, just to combine it and get what you want. So here are some results. The program is written in Go, and the default parameter were used for all the following runs. This is simply to avoid parameter tuning or trying to reduce overfitting as much as possible. So for reproducibility purpose, everything, all the experiments we run, data sets, and the scripts used to generate results are online in GitHub. So there are 3 types of data sets. One is simulated RNA-seq data. This is just simply to demonstrate this method is applicable to the existing RNA-seq data. Then we move on to demonstrate works on cfDNA data and cfRNA data. For the simulated data, actually many tools use this particular data sets to show the performance of this data. We actually borrow the data from this paper and just demonstrate this is kind of using simulated data only is kind of silly because this is we consider it's a trivial problem. If you fit this model correctly, then your performance is naturally good. This data consists of it's a 200x simulated data in 3 different rate lengths with 150 fusion events embedded. 70 of them are broken exons and 80 of them are intact exons. The broken exons, basically breakpoint is within the exons. Then 4 subsampled data from each of the subsets. You get like a different coverage for this data. Then our comparison is against star fusion and chimera scope. This is alignment-based methods, and this is alignment-free methods. So on this particular data, you can see the F-score come out beautifully. It doesn't mean this the particular methods on the simulated data is the best. It just means in the simulation, you do not capture the noise as well. The second is the cfDNA data. So the generation of this data is actually from 40 ml of whole blood. We connect in the Streck tube. Then we obtain the plasma. Then 2 stages of stage 4 prostate cancer patients. And we use the 3 healthy controls. So there are 2 fusions occurred in this data. We do not know what the true fusion is. But based on the discovery we find in this data set and based on the fact that 50% of prostate cancer contain one of those mutations, we think these are most likely fusion events. Then we expect in the healthy sample there's no fusion events. So we generate about 20 sizes larger than the typical tissue sequencing. It's about 400 million fragments. Then we have a dual UMIs. This is the results on this data. There are 2 parts of the table. It's a little bit confusing, but let me explain a little bit. On the right-hand side, this is the number of the fusion events reported by the method. So on the star fusion, it reported preliminary number of fusions. Then after filtering, this is the number of final fusion gene pairs. On the left side is basically we know these are 2 true fusion events. And in this type of data, we care about how many actual fragments supporting each fusion event. You can see like we have the preliminary and the final column for this particular data. That's just the number of fragments away. After remove the duplicates, how many fragments it turns out. So star fusion, you can see the preliminary, there are lots of genes identified, and the relatively, the filtering criteria removed the majority of them. And we don't have preliminary or, and we just have a de novo or targeted mode. You can see that star fusion in the final output, in the preliminary, there are actual fusion events being identified. This doesn't mean that we did not tune the parameter. You probably can tune the parameter for each program to get still retain the true fusion events. This doesn't mean, I want to emphasize by doing this comparison, it doesn't mean one program is better than the other. It's just you have to fit in the particular needs. Then finally, we did the cfDNA titration data. This data, it's basically a series of titration with a known fusion cell line data spiked into a genome in a bottle sample. This data has UMIs. We compare with the MANTA, which is a structural variation program. But note that we actually filtered all the structural variation excluded, but only included the fusion relevant fragments. This is a result. We have to emphasize that so the de novo mode on the DNA, cfDNA, is not actually useful. It's because you can see that the false positives just it's just overwhelming. The majority are false positive. So this is the performance, the runtime. So on the targeted or de novo mode, our runtime is actually 20 to 30 times faster. That's the star fusion, which is the only program that actually can run on our current data size. With the MANTA, this is only filtering stage at a time. But we excluded pre-processing time for the MANTA, which takes about 6 hours plus. So this is really a practical program we have developed. So of course there are limitations and caveats. I listed few of them. Because we are using the k-mer-based approach, if there are mutations near the exon boundaries, you need to consider those. Then we consider only target mode is applicable right now for the cfDNA space because of large search size. So the stage 1 output can be combined with other tools. We claim that it's not too obvious to automatically tune the parameter, which is needed. So each program needs to empirically being fit into a particular data set. I think that's all. Here are the co-authors from the company. And we want to acknowledge other colleagues from the company and Brian Haas from Broad Institute.
We just have time for one quick question.
Hi, nice talk. There's another fusion detection tool that's a similar kind of approach in which it's k-mer-based, using a method called pseudo-alignment called PISLI. I was wondering if you had any chance to explore PISLI.
Yeah, we tested that program and it come out really bad performance. I'm not sure whether they actively developed them, so we did not include in the paper.
Thank you. This is your computer.
Oh, yeah.
So we move to the next talk. Again, I remind the speakers